Hello, my name is James Hepburn and I will be taking you through the process of extracting RGB values from a series of images generated by the RZ ink test on glass. The program that I will be using to do this is a program called Fiji. This is actually a version of another program called ImageJ, but this version comes with some important plugins pre-installed, which makes it easier to use. The software is open source and completely free to use, and the website where it can be downloaded is here, and there are some basic installation instructions too. So the analysis of raw RGB data is also carried out using a macro written by Chris O'Rourke and can be downloaded from the following link. Start by double-clicking the icon for Fiji. And this small window is the main interface for the program. Click File, Import and Image Sequence, and browse to the folder containing the raw images. Select the first image in the sequence and click Open. Select Use Virtual Stack, click OK, and this will open all of the images. And you can scroll through the images using the scroll bar at the bottom of the window. I'm just going to make this smaller. So the next step is to align all of the images in the sequence. And the way that we do this is to go to Plugins, Registration, and select Linear Stack Alignment with Sift. If you leave all the settings as default and click OK. And I'm going to resize this again. And the reason we do this is because the images from the handheld scanner are all of slightly different sizes and orientations, and what the software does is look for features common to all the images in the sequence and line them up relative to the first image. And if we keep an eye on this log file here, it will tell you when it's done, which it has. And you can scroll through the aligned images to make sure that they are all aligned like so. And we can close the log window. So once the software has finished, the next step is to split these aligned images into their red, green and blue components. And the way that we do this is by going to Image, Color and Make Composite. And you can see that a new window containing the images has popped up with the red line around the first image. And another window has opened up called Channels, and this refers to the red, green and blue channels. And if I untick some of these channels, there you can see the red channel is the one being displayed. So we don't actually need this window, so I will just tick all of the boxes and close that. The next step is then to tell ImageJ what part of the image we want analyzed, and the way that we do this is by defining what are called regions of interest. If we go back to the main window and select the rectangular selection tool and hold down the Shift key and draw a box of 100 by 100 pixels in the center of the first sample. And the way you can tell the size of the box is by the numbers in the main ImageJ window, which stand for width and height. And we want a 100 by 100 box, which may not be possible. Okay, so if it's too difficult to actually draw an exactly the right size box, then what you can do is go to Edit, Selection, and Specify, and we can tell it to be 100 by 100 exactly. And if you put the cursor right in the center of that yellow box, you can move it around to anywhere in the image. But what we need is for that to be right in the center of the first sample. And we then define the region of interest by pressing Ctrl T. And you will see that a window called the ROI Manager has popped up. And if I then move this box to the next sample and press Ctrl T again, you can see that a new number has popped up in the ROI Manager, and this is basically the coordinates of that selection. So we will do that for all of the samples in the images. Press Ctrl T, Ctrl T. And you can just reassure yourself that all the samples have been done by clicking through each of the numbers in the window and checking that each sample has been recorded. So I'm just going to rename the last two samples because those are blanks just for convenience. And then if we go to the ROI Manager, select first coordinate and press Ctrl and A, you can see that all of the coordinates have been selected, and then we click More and Multi-Measure. Click OK to the pop-up, and you can see that ImageJ has returned all of the red, green and blue values from within each of those regions of interest. So if we click File, Save As, and just save it wherever you need to. So we don't need this software anymore. We have our raw data, the raw red, green and blue values extracted from the images, and the next step is then to process that raw data, and the way that we do this is by using this macro. I'm just going to open the macro and minimize it, and then open the results. And if we select all of the data, including the labels, in the results spreadsheet, and then copy by pressing Ctrl C, and go to the macro and paste or put the cursor in this cell here and paste the data. And then the next step is to click this logo, and you will get this window. Type in whatever you would like the results to be called. The irradiation interval, which would be 30 seconds in this case. The type of ink used in this case, it is Resazurin, but if you're using Acid Violet 7 or any other ink, red ink, then select that. Click continue, and you can see that the macro is actually what it's doing is calculating all the TTBs, which it has listed there. And what it also does is calculate what's called a modified Z-score, which is based on the median absolute deviation, which is basically a score of how far away a result is from the median. And you can see that based on that modified Z-score that one of these results is actually an outlier. So if we scroll down, we can see that there are actually two averages and two standard deviations given here. There's a percentage standard deviation as well. And the second average and standard deviation given is the result without the outlier. And that is the result that would be reported. So we can actually save that data now. If I go over to the right, I should also mention that the data are plotted and the TTBs are actually shown as red squares on each of the traces, and you can see that that is clearly the outlier there, data set number 7. And if we save the data by clicking this green button here, it will save the results to a separate sheet, and you can see that the macro has now been reset so that you can paste the next set of data, and the results are stored in this sheet here. I should also explain that even though this process has taken about 10 minutes for me to explain it and go through it, a well-practiced individual doing this can actually do it in under 5 minutes. So in that sense, it is quite quick.